Hello. I guess you guys probably weren't expecting this, huh? So I've decided to do a little lecture here and discuss what it is you should be studying for the exam and kind of go over things one by one. In a broad sweep, okay, the exam is going to cover chapters 1, chapters 14, 15, a chunk of, well, actually all of 20, and the very beginning chunk of chapter 23. And you should have had a reading assignment without any required homework. You know, it didn't have to do any terms or anything like that for those first 3 segments in chapter 23, which were covered on the very last lecture that we had in class. But I wanted to kind of go through this stuff and show a couple little video snippets and a few other things to kind of give you this overview as what you should be looking for and where I would focus my attention a little bit more in preparation for the exam. So starting right off, it would be nice for you to look at these sort of stories. I've said this now a few times. There's going to be a couple of questions about these stories that are in the book, especially like these ones that start at the beginning of the chapter, and then you'll find that if you look, almost every chapter works this way, is it introduces a concept, and at the end of the chapter it actually discusses the conclusion of that concept. But if we look at chapter 1, okay, so read a little bit of this, maybe jump to this, so you can kind of get an understanding of what the overall gist of the article is about. Let's just kind of go through it. It really started off with the simplest of stuff, right? We need to know that what the definition of science is. And keep in mind that science is both a process of discovery as well as a body of knowledge. Okay, you are learning science, which is the experience and knowledge that people have gained through the years, and you're learning the method of doing science. So both of those are considered science. The scientific method, okay, is something that you're certainly going to need to know. We talked about it a fair little bit. We did the bean experiment, which was kind of on the fly, talking about the scientific method. You're certainly going to need to know what a hypothesis is. Okay, I can't imagine that not being on the test. And again, this overall process. And I want to jump over to a little video snippet here. I did show that one with Mr. Anderson, which I think I have linked already someplace in the reference files. But let's jump to another one real quick. So let me pause this and set this up, and I want to show you another video that focuses mostly on the differences between the independent and dependent variables, okay, which is going to be an important concept. So hold on one second. Okay, so I like this set of videos. What's particularly cool about it is we're going to have a little summary video here, and it's going to use Mythbusters as sort of our reference point. So watch maybe the first couple of these and the Mythbusters thing, and see if you can figure out which is the dependent and independent variable.
Number 7 Science. Today we'll learn to determine the independent and dependent variable using experiments from the Mythbusters. First, let's get some terms clear. There are 2 main types of variables in an experiment: the independent variable and the dependent variable. The independent variable is the difference between the control and experimental groups of an experiment. The independent variable is the variable changed by the scientists to test the experiment's hypothesis. The dependent variable is the thing being measured during an experiment, also called data. Now let's break down some Mythbuster experiments. Experiment 1. Does adding dimples to a car increase its gas mileage? Since dimples on a golf ball are designed to help a golf ball cut through the air when hit, the Mythbusters decided to apply this logic to a car's shape in hopes that it might increase the gas mileage. Click on the link above to watch the Mythbuster experiment. Come back after to test your knowledge.
That's what I'm talking about.
By covering the clay with dimples on a golf ball that cut down its drag and make it fly further. So by covering the clay with these...
That's one dimpled car. If ever there was a car that looked like a golf ball, this is it.
It might make the car more aerodynamic.
This is by far one of the stranger things we've ever built. And to my mind, actually one of the most beautiful, and not aesthetically either. I mean, to me, the beauty here is that we've taken the phrase golf ball-like effect on the car to the furthest possible point. No one can accuse us of not going far enough. Is this treatment going to translate to better fuel efficiency? I really don't think so.
Will those dimples make a dent in the fuel consumption?
9,450 grams. That's our start weight.
Well, by now you know the drill.
Main tank solenoid is on. Let's go, dimple car. Down.
But despite 5 runs at 65 miles per hour, the clay catastrophe never comes. So it's over to Adam.
8,972 grams. All right. Time to do a little math.
What'd you get?
Okay. The clay car with no dimples was 26 miles per gallon and change. The dimpled car, 29.65 miles to the gallon. There is a golf ball like that.
I feel like eating my beret, but I'm not gonna.
That's awesome. We totally didn't expect that result.
Okay, so let's see if we can figure this out. We'll come back to this video now.
So, which of the following was the independent variable of their dimple car experiment? The dimple car, the normal car with clay, or whether the car had dimples or not? Remember, the independent variable is the difference between the experimental and control groups. The correct answer is C, whether the car had dimples or not, because that's the difference between the two groups in their experiment. Okay, can you identify the dependent variable in the dimple car experiment? Was it the number of dimples in the car, the gas mileage of the car, or the speed the cars were driven at? Remember, the dependent variable is the data collected from the experiment. Okay, the correct answer is B, the gas mileage, because it was thought that the gas mileage would depend on whether the car had dimples in it or not. Okay, let's go to another experiment. For this experiment, the Mythbusters tested to see if elephants are afraid of mice. So the Mythbusters set up an experiment where a mouse was placed under a cup of elephant dung. When an elephant walks by, the dung is tipped over and the mouse is set free. They repeated their experiment with a no-mouse dung control that was tipped over when the elephants walked by as well. Click on the link above.
Okay,
to
let's
watch
check
the
it out.
Mythbuster experiment. Come back after to test your knowledge.
Take your piece of elephant dung, and you take your mouse. This is a stand-in mouse. The real one will be later. Put them in the hole, cover them with the cardboard, lay it out. You think elephants are going to come on by, and wait in the distance with your monofilament. When the elephant gets near, beep, you expose the mouse, and all hell breaks loose.
We've chosen this area because it's open enough that we can see what's going on. And we know there's some food over in that direction, and we're hoping that the elephants will come through here on their way to the food, and at the right moment, we'll roll over the ball of dung, release the mouse, and see what happens. Hey Adam, are you done yet?
I'm all set, ready to go.
Well, with everything ready to go, all that's left is to get some fairly predictable predictions.
For me, I see this the same way I would see a person looking at a fly on the ground. It's going to be so small that you may not even notice it. I don't see any reason why an elephant would be afraid of a mouse.
I don't have high hopes for this myth.
With the sensible money on the no reaction theory, it's go time. The elephants innocently approach the dung-booth doom in what must be the most easy to anticipate outcome in Mythbusters history. But then, as crazy as it seems, the elephant backs away. The Mythbusters, like me, are flabbergasted. Working under the assumption that it must have been the movement of the dung that freaked the Ellie out, the boys reset for take 2, dung movement only. the elephant was undeterred. This is getting ridiculous.
He didn't seem to respond to the ball of dung at all, and I think my timing was exactly the same as it was with the mouse in there.
Works for me. Elephants are afraid of mice.
Not only was I totally wrong in the first test about the elephant not reacting at all to the mouse, but after it happened, I was sure that it was the dung moving. I was absolutely positive that it was the dung moving. So we do another test, the dung moves, the elephant doesn't even notice. It has to have been the mouse.
Working on the basis that science is repeatable, the boys are going to retry test 1 to see if they still get this incredible result.
All right, let's see what happens.
For the final time, the elephants approach. And lo and behold, in this David and Goliath battle, David wins again.
He definitely seemed to take notice of the mouse, like it was a significantly different thing than just the dung. I wasn't dreading this result. I'm always pleased to be completely wrong. I'm more astonished that I'm finishing the day calling something plausible that I knew wasn't plausible when I got up this morning.
He definitely didn't freak out, but he was very cautious. The mouse had his attention. It's that little grain of truth that we always look for in a myth, and there it was.
There it was indeed. So with the mouse safe and well, despite Adam's best efforts, and the elephants ready to move on, it's time to wrap this one up.
How are we going to call this one?
Well, it wasn't a stampede. It wasn't like the cartoons where the elephant freaked out at the sight of the mouse. But the idea that the elephant was cautious around the mouse, I'd say we have to call that plausible.
I agree.
What do you think?
Hello.
There you go. Let's come back to this video.
Okay, can you identify the independent variable of their experiment? Was it whether or not there was a mouse under the dung that had the mouse hiding under it, or the elephant's reaction during the experiment? The correct answer is A, because whether or not the mouse was present under the dung was the difference between the experimental groups. What about this experiment's dependent variable? Was it the number of times the experiment was repeated, the elephant's reaction during the experiment, or the number of elephants scared by the mice? The correct answer is B, because it is the reaction of the elephant that was being measured each time the experiment was carried out. Let's go to experiment 3.
Okay, so let's end this particular session. If we have time at the very end, I'll come back to these because actually I really like the next one too, which was really pretty cool. Comparing a firing of a gun versus a rock thrown from a lawnmower, which is the more deadly or potentially more deadly, which is pretty cool. So let me pause this again briefly and I'll set up the next little thing and I'll be right back with you. Alright, so as we move along, this particular section right here on page 12 I think is worth some quick review at least. I mean, it covers everything about the independent variable, the dependent variable, overall controls in an experiment, talks about placebo, which is worthy of noting again, and then of course it moves into the double-blind and single-blind experiment, okay, which I think are good concepts to review. I think that might be useful. So this section here from page 12 and 13, experiments are the gold standard for establishing causation, to right to 1.3, I think is probably worth a review. Then of course we move into the characteristics of living organisms. Obviously it's going to be on the test, okay? You know, what are the characteristics of living organisms? Let me pause this, set this up real quick, and I'll be right back with you. Okay, so continuing on, like I said, the characteristics of life are going to be really, really important. Without a doubt, I'm certain there's going to be things like that on the test. Here's a really short little video that actually kind of goes over them one more time. Let's just snap to it real quick. It's a simple one. It's 3 minutes. Let's give this guy a chance to explain to us what are the characteristics of life.
Ever wondered why we are considered a living thing? Why are we considered life? Why isn't a smartphone or computer that you're most likely viewing on considered to be alive? Why is a wooden table not considered life, be it a tree is a living thing? What is life? Well, it's debatable, but most scientists, however, do agree on some key things. These things is what determines whether something is a living being. It is called the characteristics of life. This is how we can determine what a living thing is. In this channel, Easy Science, I'll dive into the study of life, or what scientists call biology. Biology is such a diverse subject with several different topics. I will be teaching you the basics as well as detailed concepts in biology, such as the characteristics of life, which is a basic concept to understand. So, what are these characteristics? Well, for starters, all living things are made up of cells. Most of you have heard of a cell but don't really know what it is. Well, let me explain. A cell is a very small structure that makes up an organism. Basically what we are made of. This tiny picture you see here is one of many stuck together, creating a body part like a muscle or a nerve. There are several types of cells, such as the cells that make up your skin. Picture this cell among many others stuck together and stacked on top of each other. That is our skin. These organisms that have more than one cell, such as animals and plants, are called multicellular. There are other living things that are only composed of one cell, such as bacteria or most protists. These are called unicellular. Second, all living things must respond to their environment. An excellent example is a Venus flytrap, which is a plant that eats bugs. Yes, a plant that eats bugs. So basically, a Venus flytrap has these hair-like structures sticking out called trigger hairs. When an insect touches one, the flytrap will sense it and automatically close it and digest it. This is how a Venus flytrap responds to its environment. Third, a living thing will use energy to survive. What do I mean by that? Well, for example, a human's metabolism. When a human eats food, your saliva, stomach, and other digestive organs will use energy to absorb nutrients from your food. Fourth, a living thing will maintain its internal conditions. What do I mean by this? As an example, a human has a certain internal temperature to work its best. Let's say it is snowing and you need to go throw the trash outside. When you step outside, you will begin to get cold. Your body's way to fix this is by shivering. When you shiver, your muscles are moving so fast that they begin to create heat to go back to its preferred internal temperature. Fifth, a living thing must reproduce. A chicken will lay eggs, and once the egg hatches, a chick emerges. This is to ensure that a certain species does not become extinct. And sixth, a living thing will adapt to its environment. An excellent example of this is a polar bear. A polar bear lives in harsh, cold environments. The bear's way of adaptation is by growing a thick fur coat to keep itself from freezing. These are the characteristics of life. Thanks for watching.
So an important thing to consider about this video is the fact that he chose, one thing I'd like to elaborate on is homeostasis, which is the ability to maintain that control. He didn't use that term. And the other one was the last very point where he talked about adaptation. Let's keep in mind that only groups of organisms can evolve, okay? So that would be a slight different from the video that you just saw. All right, let's move ahead. The end of chapter 1 is worth looking at the summary, okay? If you don't have time to do anything else, I think it's worth it to read the summary. It does a nice job summarizing. All right, so read through this section, understand the basic concepts, grab a few of the terms, look down here's key terms, see if you can see where they fit into the summary, and I think you're probably going to be good for chapter 1. Let me skip ahead to chapter 14 and I'll be right back with you. And like I said, like most of these chapters, they start and end tying story together, okay? So it's worth looking at the finches of Darwin. You know, I've already asked questions about it. It'll probably come back to you. Okay? And of course we have the introduction of it here, leaving us with a question. And at the back of it we have evolution in action. So it summarizes it rather nicely. If we go through the book, chapter, we talk a little bit about Darwin. Worth familiarizing yourself with his name at least. Evolution and natural selection, which we spent quite a bit of time on. It's probably worth knowing what alleles are again. Okay, I think that's probably a nice key word. I will say on the last test I was disappointed how many people had kind of forgotten the importance of gene flow. Okay, You need to understand what gene flow is to be able to understand how evolution can and cannot occur. Genetic drift is probably worth a quick look. We didn't do the Hardy-Weinberg thing, and we will not in regular biology, so I don't see any particular reason for you. You can read it if you're interested. This particular section, I think we can safely skip. Okay? We have some nice examples of adaptation that are worth kind of a quick look. Remember this thing about disruptive, stabilizing, and directional selection. Okay? I think that's probably worth a quick look at as well. Keeping moving ahead. Adaptations, kind of generalized information. Sexual selection. I think it's kind of neat to know about sexual dimorphism. Okay? Things that vary between the sexes. And we see that quite clearly here with this peacock, of course. And we went into some introduction into fossils. All right? And there were some examples here. Don't forget about the Archaeopteryx, okay, that bird which is sitting there, or the bird slash dinosaur, which is sitting there right in front of the desk. An excellent example of a fossil which gives us transitional species. So we can learn a lot about the theories and support for the transition between dinosaurs and birds. Okay. Plate tectonics, please understand Pangea, okay, and the importance of how at one point in time we had a supercontinent. We have had several supercontinents, in which case animals could migrate from one place to the other without any particular problem. And then, of course, because of plate tectonics, it splits apart, and we end up with the world that we know and love right now. I think that's actually probably about it for the overview of this chapter. I think it's probably worth looking at this sort of thing here regarding the Galapagos finches. You get an idea. We've talked about it. But here's a nice illustration of the different adaptations that have occurred and their possible origins. Okay, so the common ancestor. And I love these little articles. Okay, I can't say for sure there'll be one about this one, but it's worth taking a look at this. So you do want to spend a little time going over your notes, going over the lectures and looking at it. And once again, my gosh, these summaries are really actually pretty darn good. If you throw in these key terms in the back of your head and you sit there going, oh, what is this? And what is extinction? What is a fossil? What is gene flow? And then look at the summary. You'll probably feel a lot better about yourself if you can actually kind of master this material here. Let me move ahead. Chapter 15. I think it clearly, from what I saw in the last test, a lot of people didn't really look at this section here about the Lake Victoria cichlids. Okay. And I think it's worth looking at. And again, one more time, we start with a little introductory story, but the actual guts of the article is at the very end of the chapter. Okay, so it summarizes it. Read this, and then you'll feel much better about understanding the significance of the cichlids in Lake Victoria. This is also interesting too. I think it came up in class discussion one time about whether or not there was a Neanderthal human hybrid at one point. But let's go through this real quick. Okay, definitions of species, we've seen this before. I think it's worth looking at this table down here. I presented it in the lecture at one point too, but it talks about the pre-zygotic barriers and the post-zygotic barriers. Okay, these are the sort of things that actually push along evolution, which separates species. The biological species concept, okay, I think is worth looking at. There's the morphological species concept. Keep in mind, fossils pretty much only fit into the morphological system here, okay? There's not very much, if any, DNA left. You can't look at it biologically. Whereas biological species concept, well, obviously we have a sample here we can look at. And there's some good examples here of why this is so important. You know, are these reproductive, these moths, for example, are they reproductive related or not? And in fact, there's another good example. I will find it in a moment. Here we go. Ring species. Okay, understanding what this actually really means. Why is this particular thing important? Let me go back through here again real quick. Geographic isolation, okay, is worth noting. Okay, these are all movers and shakers when it comes to evolution. And I think it's also important to notice the difference here between, let's see if I can find the keywords. Give me 1 second to pause it because I see one, not the other. Hold on. Okay, allopatric speciation. All right, look at this versus sympatric speciation. All right, sympatric speciation can occur without geographic isolation. All right, so this is a real good thing to look at about those Lake Victoria cichlids. All right, they live in the same lake, for heaven's sakes. They are not geographically isolated. And I think it's important to realize that there's more than one way and the differences that might push it. It is possible to get evolution in a single generation with polyploidy, all right? And I think I showed examples of watermelons and various other things which can occur, which can happen in a single generation, which can give us a different morphology right off the bat. Let me pause this for a second, catch up. Okay, so please take a look at adaptive radiation, okay, and understand that sort of concept, how we can have, when a niche opens up, why adaptive radiation works out so well. What happens if we wipe out if we throw a meteorite into the Yucatan Peninsula and wipe out most of the life forms? Why does adaptive radiation, why is it so powerful in that circumstance? Homologous traits, okay. Again, we can look at these particular things and see that somehow we are related because we share such homology, such common bone structures between, for example, a bat, a whale, and a human in our appendages, in this case, the arm and hand. Okay, these bones are very similar. Vestigial traits, all right? These are things that are left over. Pythons have, if you dissect them, they have little bits of their hind legs still, and even a bit of a hip there. You have a tailbone, okay? There's a number of vestigial things that are not necessarily removed from the organism, because if they're not hampering it in its development or survival, it's not a bad thing, and so they end up having this residual effect. Okay. Convergent evolution is worth noting, because again, you can have a flipper of a bird, all right, like a penguin, and we can have a flipper of a fish, like a shark, and we can have a flipper of a mammal. These things all evolved into similar structures, these flippers, because of convergent evolution. They were pushed into a common thing. And in these last couple seconds, I can't stress it enough, take a look at the summary, okay? If this is the only thing you do, read these summaries, look at these key terms, see if you can remember what they mean, tie it into the summary in your head, and you'll probably feel better. Okay, let's move on. Okay, so let's wrap this up. It's kind of amazing how time is just flying by here. So, this evolution chapter here, once again, by golly, it starts with a little story here, and it ends the chapter with sort of a nice conclusion, okay? And here, of course, is that living fossil fish. I'd love to ask you another question about that. I'm not sure I'm going to, but it would be worth looking at. And actually, what I really want to do here is point out, of course, that There's a lot of good stuff in here. We have our sort of timetable of evolution, which is probably worth understanding, and I have a short video clip on that. And I do want you to understand this is an important table here, okay? So life begins way back here at about 3.7, 3.8 billion years ago or something like that. But we really don't get anything cool happening, so to speak, until we start getting oxygen into the atmosphere, and that takes photosynthesis. And photosynthesis happens before we have the eukaryote. Remember, the eukaryote is the cell that actually has its DNA stuck inside of a ball. The prokaryotes, the prokaryote, it's before there's a ball, okay? So the DNA just floats around in the cytoplasm. And then for a long time, we just have some simple organisms buzzing around here, the first eukaryotes. We don't get multicellular organisms until, gosh, almost a billion years ago, less than that, 700, 800 million years ago. And from that point on, basically everything you know and love starts to come into play, okay? So it's worth looking at this table and understanding the importance of these main sort of events. The other thing that's kind of nice in here is this is a nice sort of section describing the various periods and the mass extinctions and let's see, where are the explosions at? I guess that's here, the Cambrian explosion, okay? Flip through these things, look at the cool pictures, look at the key terms. Once again, plate tectonics come into here because we're going to be talking about Pangea once again, okay, and why it was so important in evolution and how things were able to migrate and eventually stop gene flow, okay. And there's a talk about is there another mass extinction going on, all right. Legless lizards, we have these sort of stories here where we have that convergent evolution going on, and vestigial traits and various other things that can be turned on and off by regulating genes. And of course we have the introduction into the Linnaean system of classification, which we sort of went over really quickly. I do want to show you this cool little video though, and it'll take 7 minutes, and let's just get it going here.
Look at who our ancestors were. A simple chemical
This is an old video, but it's a good one.
In the history of our planet. There were many kinds of molecules in the primordial soup. Some were attracted to water on one side and repelled by it on the other.
In fact, it's so old that the dates are a little off.
Together into a tiny enclosed spherical shell, like a soap bubble, which protected the interior. Within the bubble, the ancestors of DNA found a home, and the first cell arose. It took hundreds of millions of years for tiny plants to evolve, giving off oxygen. But that branch didn't lead to us. Bacteria that could breathe oxygen took over 1 billion more years to evolve. From a naked nucleus, a cell developed with a nucleus inside. Some of these amoeba-like forms led eventually to plants. Others produced colonies, with inside and outside cells performing different functions. Becoming a polyp attached to the ocean floor, filtering food from the water, and evolving little tentacles to direct food into a primitive mouth. This humble ancestor of ours also led to spiny-skinned, armored animals with internal organs.
Sorry about my dog.
Our cousin, the starfish. But we don't come from starfish. About 550 million years ago, filter feeders evolved gill slits, which were more efficient at straining food particles from the water. One evolutionary branch led to acorn worms. Another led to a creature which swam freely in the larval stage, but as an adult was still firmly anchored to the ocean floor. Some became living hollow tubes. But others retained the larval forms throughout the life cycle and became free-swimming adults with something like a backbone. Our ancestors now, 500 million years ago, were jawless, filter-feeding fish, a little like lampreys. Gradually, those tiny fish evolved eyes and jaws. Fish then began to eat one another. If you could swim fast, you survived. If you had jaws to eat with, you could now use your gills to breathe the oxygen in the water. This is the way modern fish arose. During the summer, some swamps and lakes dried up, so some fish evolved a primitive lung to breathe air until the rains came. Their brains were getting bigger. If the rains didn't come, it was handy to be able to pull yourself along to the next swamp. That was a very important adaptation. The first amphibians evolved, still with a fish-like tail. Amphibians, like fish, laid their eggs in water where they were easily eaten. But then a splendid new invention came along, the hard-shelled egg laid on the land where there were as yet no predators. Reptiles and turtles go back to those days. Many of the reptiles hatched on land never returned to the waters. Some became the dinosaurs. One line of dinosaurs developed feathers useful for short flights.
There's our Archaeopteryx right there.
The only living descendants of the dinosaurs are the birds. The great dinosaurs evolved along another branch. Some were the largest flesh eaters ever to walk the land. But 65 million years ago, they all mysteriously perished.
Now we know that was a meteorite.
Meanwhile, the forerunners of the dinosaurs were also evolving in a different direction. Small, scurrying creatures, with the young growing inside the mother's body. After the extinction of the dinosaurs, many different forms developed. The young were very immature at birth. In the marsupials, the wombat, for example, and in the mammals. The young had to be taught how to survive. The brain grew larger still. Something like a shrew was the ancestor of all the mammals. One line took to the trees, developing dexterity, stereovision, larger brains, and a curiosity about their environment. Some became baboons, but that's not the line to us. Apes and humans have a recent common ancestor. Bone for bone, muscle for muscle, molecule for molecule. There are almost no important differences between apes and humans. Unlike the chimpanzee, our ancestors walked upright, freeing their hands to poke and fix and experiment. We got smarter. We began to talk. Many collateral branches of the human family became extinct in the last few million years. We, with our brains in our hands, are the survivors. There's an unbroken thread that stretches from those first cells to us. Let's look at it again, compressing 4 billion years of evolution into 40 seconds.
Okay, I thought that was actually pretty good. And if you just kind of imagine in your head or try to retain that sort of imagery, it'll help you answer sort of questions about what came what and when and where and why. Let me pause this and I'll set up this next section. Okay, so finally we come to the end of it all, and it's going to be the newest material. Biomes, I think most of you are actually pretty much set for. A little bit of it about ecology. I would say the biological hierarchy is nice to know, starting with organism, population, community. Take a look at this, see if you can understand this general concept. Don't forget, at some point in here we talked about biotic and abiotic. Abiotic are the non-living components of an environment, and the biotic are the living components. We did a brief little thing on climate, which is the most important factor in determining a biome, and it's nice to kind of get an overview of, I can't imagine asking anything too deep about this, but understanding the various cells, the convection cells, and why we have these different zones. Okay, so we have the heat rising, which is causing the various changes in pressure, and it's falling back down. And we have the rotation of the Earth with the Coriolis effect, which is causing the trade winds. Okay, rain shadow, I briefly talked about, which is when you actually have the rain forming because the air is being physically pushed high where it's colder and it's condensing, and it's causing it to rain. the biomes. We played the game. I think the only thing that's worth noting, per se, is Chaparral was not in the game at all. It's probably worth looking at that one. And of course, knowing the other ones as well. But I think you basically have that. I got the sense that most of you have already done this kind of material at least one point. Aquatic biomes, the marine and freshwater biomes. I talked about estuaries. I love estuaries, super important things. Don't forget about my little discussion about the sponge, how it helps absorb the changes in floodwater and conditions. You guys have that nice chart of the marine biome. I hope you have it complete. Okay, that illustration that we did in class when I did that presentation, I handed you all a chart. Kind of get an idea of the different zones. Okay. See if you can figure out how to label those zones. Don't forget about it. Take a look at that map. I think that's basically it for 20. Let me pause it and do this quick little thing about the last of it, and then that'll be end. So hold on a second. As usual, I can't stress enough, these summaries are pretty nice. Okay, take a quick look at it. If that's the only thing you do, that's probably what I would take a look at. And then we ended up with 23, and it only went to the first couple of sections and went up to 23.2. Yes, 23.3 is as furthest as we went. We did not include that. And it talks about the relationships, okay, that happen in a niche or an ecosystem. And I think it's worth looking at this as well. This might be new for some of you people. Mutualism, symbiosis, certainly going to be key terms to look at. Pollinators, commensalism, and we had exploitation in here. Okay, so take a look at these things, parasites, and you know, I don't know what else to say. I'll probably put some more material up on the web over the weekend. On Saturday, I might throw a couple additional things up there for you to have as a reference point. And that is it. So good luck. I'll be back on Monday, and I'll see you all soon. Bye.